All right. I can see my gear over there. That's all right. Okay. Looking to see if anybody's going to join me here in a second. So we're doing our review for test 1. I'm going to actually pull that test up so I can take a look at it so I can really confirm what you guys need to know here. Hi, Willow. So I'm in my office in my house, and I was supposed to set up my OBS, system, but it didn't work out that way. I just got home, so yeah. I just walked in the door. So let's see here. Let's see. We got Bio 1140, and we're just, not spring. That was. Then we got fall 22, and we got exam 2. I'll pull that up here. Hey. All right. So as we get going here, the best way this is going to work is for you guys to basically shoot me a top chat here and let me know what your questions are. But I can tell you just hi, Willow. My daughter's coming in here to join me. And here comes the wife. Goodbye. Oh, and you get to see my dog here. He's in the background as well. Yeah, we're amateur hour over here. That's why I'm building an entire studio. Okay, exam 2. Why won't it pop up? All right, as we get going, you guys have any specific questions here? What in the world? Why is that exam not showing up on me here? Okay, this looks like we're getting more and more people here. And of course, for whatever reason, my word does not want to show up. Well, I can see it down there. Oh, stupid. That one's available. All right, file. We'll go to it. So nobody's hitting me up with questions. So I will tell you for this test, this is some of the stuff that really people have a hard time with. And here we go. I got it now. Yeah, so what people have a really hard time with on this test, and I've been teaching this for, oh my gosh, better part of a decade. And for whatever reason, that idea of, or the concept of bonding is very hard. Covalent bonds, hey, you're just sharing electrons, right? If you're sharing them equally, you've got nonpolar covalent bonds. If you're sharing them unequally, you've got polar covalent bonds. If you have nonpolar covalent bonds, you are a hydrophobic molecule like gasoline, like olive oil, like fats, and you store a lot of energy. If you have polar covalent bonds, you store less energy. Those bonds are stronger, and you can often form hydrogen bonds with water, and you are hydrophilic, which means you can dissolve in water. Okay, now I've already got a question here. What are some highlights of each chapter I need to study for Wednesday's test? Hold on, I'll come back to you because I'm right here talking about why are nonpolar molecules hydrophobic. In a nonpolar molecule, let's see here. I will draw one out for you if I have a pen here. Let me draw you a molecule called a fatty acid. Actually, I'll just go ahead and draw a molecule of fat for you. Now I'll just draw this right here. It's got 3 carbons, some oxygen, and we can explain these things here like this. Okay, so polar molecules. So I don't know if you guys can see that right there. Okay, what we've got here, this is a molecule called a triglyceride. This is fat. Up at the top, that's carbon. This line represents a covalent bond. That's carbon. These lines right here, this is actually on the test. I got an arrow pointing to this right here. And I said, what is this? And you're going to say non-polar covalent bond. And right there is a carbon. And the reason why non-polar covalent bonds, like if we draw the molecule methane, this methane molecule right here, just carbon, hydrogen, there's our non-polar covalent bond. There's no way for that molecule to form a hydrogen bond with water. Of course, water looks like this. It's H2O. Here's our molecule of water. Water, of course, is hogging the electrons, so it becomes partially negatively charged. The electrons pull away from the hydrogens. This becomes partially positively charged. So now we got a minus and a plus end. Okay, a minus and a plus end. There's the minus end, there's the plus end. But this is methane over here. There's no minus and plus ends. There's no way for this to interact by attracting to this water. So it's hydrophobic. It'll actually float on top of the water. But if I have another water molecule here, this is one water molecule. Here's the other one. This hydrogen is covalently bonded to this oxygen, so that's water. But these little dots right here, that is our hydrogen bonds. That's just an attraction between this positively charged hydrogen, it's partial, and this oxygen here is partial negative. They attract. This molecule right here, it's all carbon and hydrogen. It's hydrophobic. There's no way for it to interact with water. That's why fat, like olive oil or bacon fat, floats on top of water. And also, because oxygen loves these electrons, it holds on to them very tightly. This is a hard bond to break. So you throw a match in a water, nothing happens. The match goes out. It lacks energy. It lacks the activation energy to break this molecule. This over here, hydrophobic, sharing those electrons equally. Those bonds are easier to break. They store more potential energy. You throw a match in gasoline and, well, you know what happens. It goes boom. Okay, so that's what you need to know. Also about the chapter 4, the macromolecules, because I know that somebody is asking about what they need to know for each of these chapters. In chapter 4, we talked about 4 macromolecules: carbohydrates, proteins, nucleic acids, and the lipids. And on the very front page of the test, I have an amino acid, a carbohydrate, a nucleotide, and a triglyceride, in that order across the page: A, B, C, and D. Yeah, you guys better write that down. And what I'm going to do for the next 5 questions on that very front page, I'm going to ask you questions about those four molecules: the amino acid, A, the glucose molecule, B, which is carbohydrate, the nucleotide, C, and the triglyceride, which is a fat molecule, D. Okay? You need to be able to recognize which one builds proteins, which one is used for energy, which one is used to store information, which one stores the most amount of energy, right? And of course, fat molecules store the most amount of energy. Nucleotides store information. Amino acids are used to make proteins, and carbohydrates can be used for energy inside of cells. So these are the types of things you need to know about these macromolecules from chapter 4. What do they look like? What do they do? That's important. Okay, let's see here. So that's the important part for chapter 4. You got to be able to recognize those macromolecules. You've got to be able to do more than say nucleotides build DNA. You need to know what a nucleotide looks like. Now, I don't have you memorizing adenine, thymine, guanine, cytosine, and uracil. I'm not having you do that in this class, but you need to be able to recognize that a nucleotide has a 5-carbon pentose sugar attached to a phosphate and attached to a nitrogenous base. So there's 3 things there that make up a nucleotide: the sugar, the nitrogenous base, and the phosphate group. And all nucleotides have that. So that's really important for chapter 4. The next one, we got the organelles. And we got one: what are the most important concepts regarding organelles? Okay, you need to know the difference between the endomembrane system and the energy converting organelles at a first pass. So the energy converting organelles, mitochondria and chloroplast, are not part of the endomembrane system. That's a test question. All right, you're going to need to know that. The endomembrane system is found only in eukaryotes, and they are made up of the nuclear envelope, which is these membranes surrounding the nucleus. the endoplasmic reticulum, of which there are 2, 3 different types, depending if you're looking at muscle cells. Smooth ER, rough ER, and sarcoplasmic ER. So in the endoplasmic reticulum, the smooth makes like lipids and is used for detoxification. The rough ER has ribosomes attached to it. Ribosomes are used to make proteins, so the rough ERs can be involved with protein synthesis. And for those of you going on into AMP or studying muscle physiology, the sarcoplasmic reticulum that's in the sarcoplasm, that's unique to a muscle cell, and it holds calcium. So endoplasmic, so the endomembrane system, the endoplasmic reticulum, the Golgi bodies, various transport vesicles, and also the vacuoles as well, and the lysosomes. Those all form the endomembrane system. They are a series of membranes that are either directly connected to each other or indirectly connected to each other. What we mean by indirectly connected is that you can have transport vesicles will carry proteins from the endoplasmic reticulum, specifically the rough ER, to the Golgi body, and then proteins can exit the Golgi body and transport vesicles to be exported outside the cell, to be embedded inside of another membrane elsewhere inside the cell, or to be inside of an organelle. Okay, another thing that's very important for cellular organelles. Remember that we have 2 types of cells: prokaryotes, eukaryotes. Prokaryotes lack mitochondria. They lack an endomembrane system, and they lack a cell nucleus surrounded by a nuclear envelope. There's no cell membrane surrounding a cell nucleus inside of a prokaryote. So prokaryotes are typically smaller and lack the internal structure of eukaryotes. That's important. Now, prokaryotes include the bacteria and the archaeans. Eukaryotic cells include plants and animals and fungus and many types of algae and single-celled organisms. Plants are not prokaryotes. They are eukaryotes, and they have chloroplast and mitochondria. Yes, you'll see that as a test question. Plants have mitochondria. They do cellular respiration because they are eukaryotes. So when you see questions regarding differences or similarities about cells, if I say all cells, I mean both prokaryotes and eukaryotes, of which I will have in parentheses. If I say all cells, I'm going to say prokaryotes and eukaryotes. They might have something like, they might share things in common like DNA, genetic codes, same amino acids, but they don't share in common mitochondria, endomembrane system, or a cell nucleus surrounded by a membrane. Let me say that again. Prokaryotes and eukaryotes, all cells, share many things in common. From cell membranes, they share in common the same genetic code. They use the same 20 amino acids, all right? And they have many of the same metabolic pathways, like glycolysis. What they do not share in common, prokaryotes and eukaryotes do not share in common. They do not have mitochondria. Prokaryotes don't. Prokaryotes don't have that cell nucleus. They don't have the organelles that the eukaryotic cells have. And I'm going to say this one more time. Plants are eukaryotes. Okay, so that is the most important concepts regarding the organelles. And also when it comes to the mitochondria, mitochondria have lots of functions inside the cell. They do more than just make ATP. They've been incredibly important for the evolution of eukaryotes, including the evolution of plants and animals. We basically could not exist today without mitochondria. Okay. All right. Essence Copeland. Where can I find the chemical structure of the macromolecules? Like what slide? Well, let me see here. That would be chapter 4 there. And we can pull up chapter 4 and I'll give you some guidance here on that. Let's see here, Bio 1140. Yeah, too bad I got to figure out how to do screen sharing on this thing. I had it all set up in my office studio here, and then got a little late start. All right, so chapter 4, macromolecules. So basically, let's see here. Macromolecules. Well, if you carbohydrates, let's see here. I have a picture of let's see, carbohydrates. Let me go back and forth here. Yes, carbohydrates. Slide 32 shows a glucose molecule. And then when you see a complex carbohydrate on page 42, you see a saturated fat on page 50. You see a phospholipid on 63. You see cholesterol or testosterone and estrogen on 66. You see the lipoproteins, HDLs, LDLs on 68 through 70. There is a question on the HDLs and LDLs on the question. And let's see here. The general form of a protein is on 81. And then the general form of the nucleotide is on slide number 98. So that's the actual slides that those macromolecules are on. So I know you guys have them, and they're also in the required reading as well. Okay. Let's see here. Somebody got their message retracted. Macromolecules, polar molecules. And then, okay, so we got another one that says some highlights of each chapter that I need to study for Wednesday's test. Okay, so we did the macromolecules, right? You have to be able to recognize an amino acid, a glucose or a carbohydrate, a nucleotide, and a triglyceride, a lipid molecule. And you need to know like which one stores the most in, which one is used for storing information, which one makes proteins, which one has the most amount of energy in it. Those are the types of questions that I'm going to ask. Because it's more than just, can you say nucleotide makes DNA? Can you recognize a nucleotide? It's more than just saying lipids make fats or triglycerides make fats. Can you actually recognize it and know why it stores why it stores energy? Okay, then let's go on to this one. This is a good one. When we go to chapter 6, some highlights. There's a whole bunch of topics in chapter 6 that are all related. You need to know what energy is. It's the ability to do work. You need to know that membranes basically are the barrier to our systems, and membranes are made up of phospholipids, and they exhibit selective permeability. So only certain things can cross a phospholipid in or out of a membrane without help. So small hydrophobic molecules, they can diffuse into or out of the cell. So example, when you're breathing, your cells are using oxygen. So oxygen is constantly being used inside your cells. So oxygen will be diffusing across the membranes into your cell. As you break down carbohydrates, lipids, fats, proteins, you use up oxygen, but you release CO2, carbon dioxide, and that diffuses out. Cell doesn't have to do anything to do that. However, to move something like electrolytes, sodium, potassium, chlorine, calcium, or chloride, I should say, those are electrolytes. Those are charged particles. They're also ions. The cell membranes, because of their hydrophobic interior, really forms a barrier to the movement of these ions in or out of the cell. So in this case, the cells either have to create some type of channel protein that can allow them to passively pass through that protein. It's called a channel protein, like an ion channel. And that way you can facilitate the diffusion. Okay, cell doesn't have to spend any energy doing this, but it does have to create like a protein for it to flow through. Now that's just flowing down the concentration gradient. Lots of times cells have to actively pump ions against their concentration gradient. So for example, our cells constantly pump sodium out and pump potassium in. In that case, that requires a source of energy like ATP and a proton pump to move it against this concentration gradient. That's like moving a ball up a hill. You need to like use some energy to do work and something to actually physically move the ball up a hill. Same goes for active transport. Now, coming back to diffusion one more time. Diffusion requires energy. It requires kinetic energy, the energy of motion. If you had nothing moving around, right? If nothing was moving around, then you're not going to have diffusion, because diffusion is a movement of a substance down its concentration gradient. So if there's no kinetic energy, there's no movement, there's no diffusion. But cells do not have to add energy for diffusion. Think about putting a drop of water in a glass, a drop of dye in a glass of water. Think about that. Do you have to stir the water? The answer is no. The dye will eventually spread out and will go all the way to equilibrium on its own. That is diffusion. You don't have to stir it. If you stir it, it just goes faster. So remember that. That's important. Energy is required for diffusion, but cells don't have to spend any energy doing diffusion, and that goes for facilitated diffusion as well. But facilitated diffusion requires a transport or channel protein. Okay. The other one is no energy. What it does, it's like the ability to do work. There's stored energy. There's kinetic energy and other different forms. For us, potential energy is very important, because and chemical energy is potential energy. That's the energy stored like in the foods we eat, and we transform that energy and we transfer it to the energy currency of life known as ATP. And because life is a sum of all these chemical reactions at the cellular level, to make those chemical reactions, we require enzymes, and enzymes speed up chemical reactions. And the reason why chemical reactions don't go on their own is because of what's called the activation energy. And the activation energy is why you can have natural gas out in your propane tank and it doesn't just explode spontaneously, or why you don't do spontaneous, why you won't never explode spontaneously on South Park. Oh yeah, I'm still wearing Eric Cartman here. You were talking about respecting authority. But right here, this would be the activation energy. So when you're starting a fire, this is your spark, this is your products. This is your reactants, this is your products. So you've got to like spark, you got to add energy here to break this activation energy, and then your products will form and they have less energy. And of course, this is the amount of energy released, and that would be an exergonic reaction. So enzymes help lower this activation energy to get the reaction to go forward. You'll also need to know in chapter 6 about a redox reaction. Redox reactions are basically the movement of electrons. And as you're gaining electrons, electricity, energy, got the connection. If you're gaining electrons, you're gaining energy, you're being reduced. If you are losing electrons, you are losing energy and you are being oxidized. So this reaction right here, carbohydrate, glucose plus oxygen, yields carbon dioxide plus water, and it releases energy. This is an exergonic reaction. This is cellular respiration. And what happens is this carbon is being oxidized to carbon dioxide, because the oxygens are pulling the electrons away from the carbon. So the carbon is being oxidized, the oxygen is being reduced. Okay. And that is also very important, because our cellular respiration is a controlled redox reaction. And photosynthesis is also a redox reaction. It's the reverse of this, where you're taking carbon dioxide and adding the hydrogens onto it to make glucose. And this would be the carbon getting reduced in photosynthesis. And that's an endergonic reaction that requires an input of energy. Okay. Let's see here. Is kinetic energy already present in facilitated diffusion? Yes. Great question. Does active transport use kinetic energy from cells, or does it require an additional source of energy? Another fantastic question. Active transport requires an input of energy, usually in the form of ATP. You can also use electrons too. We'll find out next chapter. Okay, I just went over redox reactions. Let me know if I need to do that again. And then how do non-polar covalent bonds store more energy than polar covalent bonds? I thought polar covalent bond had more energy because the electrons aren't equally shared. Okay, that's I understand what you're thinking. That's actually a really good question. And yeah, so we won't get, I got to remember, I'm getting ahead of myself here trying to draw molecules. Okay, I'm going to draw water and methane again. Methane is a very, very simple organic molecule. So here we are, water and methane. Non-polar covalent bond, polar covalent bond. The energy in a covalent bond is a function of the distance of the electrons from the nucleus of an atom. So the further away electrons are, the more energy they have. The closer to the nucleus, the less energy they have, because the nucleus is holding on to those electrons, right? Like charges or opposite charges attract. Those protons are holding on to the electrons and keeping them from moving around. But as they get further away, they can move around more, right? So oxygen here is going to hold on to these electrons. It's going to hold them in place. So they have very little energy. And that makes this molecule stronger. This, on the other hand, similar electronegativities. The electrons, instead of being close to the oxygen here, they're equidistant between the hydrogen and the carbon. And because they're equidistant, they're as far away from the nucleus as they can. They have more potential energy. All right. So yeah, chemists have spent like lifetimes figuring out all those bond energies, which we don't need to concern ourselves with here. It's just understanding at that level explains like why carbon dioxide, why water have very little energy, and why you need energy from like sunlight or some source to break them apart, to break those bonds and reform them into like glucose molecules like you get in carbon dioxide. And it also explains why you take something like carbon dioxide or fats and why they have a lot of energy. And when you break them apart, it takes energy to break those bonds, but a lot of energy is released when those bonds set. Okay. And can you by chance explain what we need to know about endosymbiosis? Fantastic question. One of my favorite things to talk about. Basically, endosymbiosis is the origin, we think, of eukaryotes. And we have very compelling evidence to show that two prokaryotes, a bacteria and an archaean, merged. The bacteria bored its way probably into the archaean. And from there, that bacteria evolved into the mitochondria. In fact, the mitochondria to this day still have their circular DNA, just like bacteria do. And so that was basically the origin of eukaryotes. And that's the most important thing to know about endosymbiosis. And for evolutionary purposes, it explains why eukaryotic cells are really similar. I know you and a plant cell are way more similar than you think. And it has to do with that single origin of eukaryotes about 2 billion years ago. And that's the most important thing to know. That so we talk about the origins of eukaryotes. It's through that endosymbiosis. And it's not like some prokaryotic cell like a bacteria slowly gets more and more and more complex and evolves into a eukaryotic cell. That's not how it happened. It started with the merger. That's actually a test question too. All right, hey guys, thanks for your likes, man. I appreciate that. Makes it worth it for coming out tonight to help you guys out. All right, do you guys have any more questions for me? I want to look at this test one more time here and see what else you need to know. Oh yeah, know what happens when proteins denature and why that's bad for you. Know which of the following, know which lipids are unhealthy. be able to recognize something that's hydrophobic versus hydrophilic, the energy currency of life. Okay, I talked briefly about bacteria diversity. You need to know like just something about them. Are they primitive? Remember, I don't think that they're very primitive. And the reason why they get called primitive is because they're small and they lack all the organelles. But the reality is they evolve very, very quickly. Okay, so you need to know that. I see some more questions pop in. I'll come back to that here in a second. You also need to know about the cytoskeleton for chapter 5 as well. You need to know the actin filaments, the keratin fibers, and the microtubules. And you need to know that like the actin filaments are very important for muscle fiber. You need to know that like microtubules are very important in like cell division, which would be called mitosis, pulling the chromosomes apart. Those are important. Let's see here. You also need to know why it's important to study mitochondria. And just remember, they're more important than just making ATP. One of the things that we'll talk about in chapter 7 is that the mitochondria are at the heart of our cellular metabolism, right? And they also just made it totally possible for all life to evolve. Also on chapter 5 about lysosomes, we study these organelles, and a lot of times we get in these intro classes and we're like, oh boy, I'm memorizing all these parts of the cell. But it's really understanding those parts of the cell has a lot of implications. We can understand aging. Hold on one second. We can understand aging by understanding how mitochondria accumulate defective copies of themselves, or how different species use their mitochondria differently, or how lysosomes fail to turn over the cellular organelles. The cell biologists call it autophagy, but it's autophagy, right? I don't know why they have to pronounce it autophagy. Yeah, so that's important there. Passive transport, active transport. Yep, got all that. Photosynthesis. Know why cold water fish are healthy for you. They have polyunsaturated fats. Oh, you also need to know osmosis. Osmosis is a special case of diffusion for water. And basically, water moves from high potential to low potential. As you add salt to water, you lower the potential because the salt, which is sodium and chloride, disassociates their charged particles, the sodium and the chloride, and they hold on to the water and prevent it from moving around. All right. And yeah, that's a pretty good one there. Okay, you got another good question here. Can you explain why high entropy can't do work? Okay, so energy is the ability to do work, right? I kept this really simple for us, but basically there's something called free energy, or Gibbs free energy, which is the ability to do work. And every time we use energy, that Gibbs free energy declines as this property called entropy increases. High entropy is a system at equilibrium, or a system that has high disorder. The best way to think about this, why high entropy can't do work. Right now, I'm in my office. It is high entropy, right? I mean, you guys can see all my gear back here. I'm getting ready to move out of this office into my big office. This one's 9 by 13. My new office will be 12 by 34 feet. So 9 by 13 into 12 by 34. I'll have a little bit more space. But you can see high entropy, right? Actually, I know where everything is at, so it's not total entropy. But think about the air in like the room you're in right now. Every air particle in that room is moving hundreds of meters per second. Wow, fast. I mean, it could go across your entire building in a second, but they don't do that. They're constantly colliding with each other, and they're moving in completely random positions or random directions. And by doing so, the air isn't moving. And if you notice in a room that was still air, there's nothing. The ability to do work is to move something. So if the air is moving, that's high entropy. The air is at the same temperature at the top And yeah, it's an equilibrium. Now, those air particles move in one direction, or I have all the molecules in a concentration gradient going from high to low, diffusing in one way across your membrane, oxygen moving across your membrane, sodium flowing into your membranes through a facilitated diffusion. That is not random. That is going in one direction. That's like a breeze. A breeze can sail a boat, turn a wind turbine, generate electricity, move branches. If it's so when they're all moving in one direction, that's low entropy. We have free energy, gives free energy to do work. But once we use it, it goes back toward equilibrium. The energy's still there. You just can't harness it because they're all moving in different directions. It's like trying to get cats to pull a sled. Good luck with that. The cats all just go in different directions. Okay, Essence, I hope I answered your question. Chapter 5, what do I need to do? Study well on the test. Yeah, make sure you memorize your organelles. Make sure you memorize what they do. Make sure you memorize also the parts of the cytoskeleton we talked about. The different functions of microtubules, actin filaments, and keratin fibers. Yeah, remember the microtubules. Those are the hollow tubes. You can push and you can pull with them. Microtubules help give the cell shape. They help push the cell apart during mitosis. They help arrange the organelles. They take the chromosomes, move them to the center of the cell, and then pull the chromosomes apart. That's microtubules. Actin filaments, that's muscle, right? So that's like your contractile protein that forms your muscles. Every movement in your body is used by some muscle contracting with actin and myosin. So actin is very important for that. Okay. Let's see here. And then keratin fibers, those are more permanent structures of the cell. They're also the intermediate fibers. They form things like fingernails, hair. And when you see dead skin flaking off of you, what you're seeing mostly is keratin fibers. Oh, okay. Essence, what did the double line mean between the C and O in a triglyceride molecule? We got a future biochemist here. Okay. We got our triglyceride right here. What this represents, this double bond right there. If you remember, oxygen has 8 protons, 8 electrons, 2 electrons in the innermost shell, 6 electrons in the outer shell, 6 valence electrons. It wants 8. So it needs to share 2 pairs of electrons. And there it is, sharing. Each line represents a pair of electrons. There's your 2 pair of electrons right there. Carbon, element number 6. So it's got 4 valence electrons, wants to form 4 covalent bonds. There they are. 1, let me take my pen off, point right here. 1, 2, 3, 4. So this double bond right there in the triglyceride, all I'm doing is forming 2 covalent bonds. So excellent question. Okay. How are ribosomes involved in photosynthesis? That's a really good point. That's something else I forgot to mention. Remember this, a common feature of all cells, prokaryotes and eukaryotes, ribosomes make proteins. Ribosomes make proteins. Now, in photosynthesis, we have the chloroplast is where we make or where we do photosynthesis. However, the ribosomes are actually involved in photosynthesis because photosynthesis requires a lot of different proteins. And guess what? The ribosomes help make the proteins used inside the chloroplast for photosynthesis. But I don't know if you meant the chlorophyll or the chloroplast. But yes, ribosomes are definitely important for photosynthesis for making all of the proteins required for it. Okay, do we need to know any specific information about the diversity of eukaryotic cells, like the difference between diatoms, and no, you do not. I don't have anything on there. I'm glad you asked that, so we can like narrow it down some of what you have to study. But there's no questions in there about the diversity of eukaryotes. All right, guys, how are we doing? Any more questions here? You guys have been asking really good questions. I'm going to check email really quick, make sure nobody shot me an email while we were waiting. So I'd like to make sure I answer everybody's questions. Okay, here we go. Somebody just sent me a whole bunch of them here. What does a nucleus? Okay, so somebody actually really sent a bunch of questions. I'm going to take a second here and go through them for you guys. But basically, I think this person is asking, what's inside the nucleus? Is it just DNA? You guys don't really need to know this, but it's really interesting. The cell nucleus is one of the most amazing structures in the world. Our brains are, of course, but the cell nucleus can seriously organize like 3.5 billion base pairs of DNA, copy it in a few hours, open it up, go to one specific place of it, transcribe the messenger RNA to make a protein. So inside the nucleus are all kinds of DNA, RNA, and lots of proteins to help fold and regulate the DNA, copy the DNA, make protein, make messenger RNA. It's incredibly complex. What's actin filament structure? Actin filaments are made of actin subunits, so you can build them up and take them back down. The Golgi is part of the endomembrane system, and it's used for like a packaging of the proteins. It's like if you think about like if you're making a car, you would machine the main body of the car with the ribosomes attached to the endoplasmic reticulum, the rough ER. And then you would take that shell, and when you would go to the Golgi apparatus, you put the finishing touches on it. Give it a paint job, give it the new car smell, install the upholstery, put in the radio, put in the speakers, kind of give those final touches, and then you ship it off to the dealership. And inside the cell, of course, the Golgi will then ship the cell. Okay, and they asked a really good question for you guys. Not only what is the Golgi used for, what is it made of? Every organelle inside of a cell is considered a membrane-bound structure. So that doesn't mean it doesn't mean you could have the cell nucleus, the endoplasmic reticulum, Golgi bodies, mitochondria, chloroplast. Mitochondria and chloroplast are not part of the endomembrane system, but they still have membranes, lysosomes, peroxisomes. All these membrane-bound structures are all surrounded by cellular membranes, which are phospholipid bilayers. All of them. Okay. All right. I already talked about that. Source of long-term storage energy. So the long-term source of energy for storage, of course, is fats. We know this. If we start eating too much ice cream, drinking too much Kool-Aid, beer, whatever, what happens is we take that excess carbohydrates and fat and we store it in what's called adipose tissue, which are fat tissues. We store it as lipids. And the reason why, think about gasoline. Gasoline is octane. 1, 2, 3, 4, 5, 6, 7, 8. And our lipids might have 12, 14, 16, 18 carbons long. They store way more energy than a carbohydrate because carbohydrates have hydrates, oxygen in there. So sugars have less potential energy, gram for gram or pound for pound, than lipids. So lipids are a long-term source of energy. Okay. Let's see. What macromolecule store the most energy? So the macromolecule that stores the most energy, you guys know this, it's a lipid. Cellular membranes, and what structures are they? It's a phospholipid bilayer. Remember, it's made up of those fatty acids, a glycerol, and a phosphate, so they're amphipathic. Protein enzymes, we covered that in class today. Protein enzymes, those are proteins, and enzymes help speed up chemical reactions. Okay. Talked about nonpolar and polar endergonic reaction. What are differences in the products and reactants? That's actually a good question. Endergonic reaction, that requires energy. Photosynthesis requires energy. So photosynthesis is where you are going to take some carbon dioxide plus some water, and some source of energy, and we are going to reduce the carbon into an organic molecule. So this is photosynthesis. This is taking carbon dioxide plus water plus a source of energy, and we're going to take this carbon right here, and we're going to add the hydrogens to it and the electrons from this water molecule to make glucose. And this is the product. This is the reactants. The reactants have very little energy, and you know this because if you light a match on any of these things, nothing happens. But you can actually burn. This is what makes paper. You can burn this. So the products here store more energy than the reactants. Now, if we switch this and go this way, and I take the glucose molecule. Oh gosh, I made a mistake. Nobody said anything. Okay, O2. Sorry, 6O2. Getting ahead of myself here. Now, if we go this way and we react the carbohydrate to the oxygen and we release carbon dioxide and water, in this case, the reactants have more potential energy than the products. So this is going to release energy, and that would be an endergonic reaction. Okay. I think that does it. I haven't seen any other information here. Hey guys, thanks for showing up tonight. I haven't seen any other questions. Are we good? Are we good? I guess so. It looks like a couple of you still on here. Well, I'm gonna call it. I appreciate you guys coming out tonight. Good luck on the test here. What do we need to know about the citric acid cycle? Nothing for this test. We're going to get into that on chapter 7, which will be on test 3. So I'm gonna talk about the citric acid cycle a lot. We're not going to go into like all the details of the chemical reactions of it. That's for you guys. That's not important. What I think is very important is realizing that the citric acid cycle is like the heart of living organisms. It's like the heart of life, and life probably began with the citric acid cycle because it's like this, just the center of everything our cell does. And to me, that's just a fascinating thing that is just shared across all life. All right. I will see you guys on Monday. This time I have the test being delivered to my office on Tuesday, so I should be there early. And I will also be in the classroom early so that I can start handing out the test early. We also don't have a class in there afterwards. So if you guys need a little extra time on the test, I think we can do that. We did last time. And like I said, I'm going to try to get there early. All right, you guys, you are more than welcome. I'm happy to do this. We will continue doing reviews. If you want to keep doing these in the future, we can do them once a week or once every other week or so. This is pretty easy for me to do for you guys. And it seems to be helpful. A lot of you showed up. All right. I'll see you Wednesday.